to the previous speaker, I don't think I could have had a better introduction. I'm here to talk to you about the focus of my lab, which is getting large molecules out of endosomes. How are we doing that? Well, compartmentalization is a thing. Once upon a time, about 1,000 years ago, I was a postdoc. And during that time, I studied the molecular regulation of vesicle fusion pertinent to lysosome biogenesis. Oh, that was fascinating. It was a lot of hard work. I learned a huge amount. But I also taught and studied human gross anatomy. So yeah, taking apart dead bodies was a thing, but this wasn't some sort of strange fetish. It was actually me learning about human gross anatomy in order to teach it to students. During that time, I became aware that your body is composed of sheets of cells folded into tubes or balls. And why does your body do this? It does this to compartmentalize, and it compartmentalizes to allow the simultaneous execution of mutually exclusive processes, such as anabolism and catabolism. So you have your stomach and you have your cranial vault. What do you think would happen if the content of my stomach, which is busy digesting my glorious breakfast, got into my cranial vault? Well, the talk would end fairly quickly, and it may amuse you, but I don't think I would be very well. Now, this is an example of compartmentalization on the gross level. However, it also happens, how do I advance? Wonderful. It also happens at the level of the cell. So these are epithelial cells. There's two of them here. This is actually my desktop. Yes, spot the nerd, that's me. You can see nuclei here, and you can see these little vesicles. These are late endocytic structures that are full of red wheat germ agglutinin and also have been immunostained with an antibody against lysosome-associated membrane protein 1, LAMP1, which is a marker for late endocytic structures, including late endosomes and lysosomes. And this is an example of the cell compartmentalizing. You have the cell's stomach, which is the endolysosome, and the cell's cranial vault, which is the nucleus. Never the twain shall meet. And from this compartmentalization, we can draw a conclusion that A does not equal B, where A equals the outside of the cell and B equals the cytosol, which are separated by membrane, the same membrane that forms the barrier function of your vascular system, because that is actually membrane forming the lumen of your blood vessels. Okay, still plasma membrane. A function of this, or the functional consequence of this, is seen here. This is a large molecule, a protein, bovine serum albumin, which we've chemically labeled with Texas red, a very, very powerful fluorophore. And we've chased this BSA in the presence of a protease inhibitor, lupeptin, into a cell. And we've labeled those cells immunologically with either labels to an early endosomal marker, EEA1, or a late endocytic marker, LAMP. And you can see these little red dots, which is the protein, inside these rather glorious green donut-like structures. This is the compartmentalization of our bovine serum albumin. And another consequence of the barrier function of these little green donuts, our late endocytic structures, is that not only can they compartmentalize proteins, they can digest them as well. And if you look at protein toxins, which, like viruses, have evolved to move large molecules over barriers like membrane, these protein toxins often have a cell trafficking and cell binding chain and a warhead, which is the catalytic chain, which actually mediates intoxication after its delivery to a specific compartment. So if we take a fairly well-known, notorious toxin, ricin, and we put the holotoxin on the cells die fairly quickly. Now, if we remove the cell trafficking chain, the B chain, and just give the cells the warhead A chain, you can see there's up to about 4 or 5 logs difference in toxicity. 4 or 5 logs, that's a lot. And you can see this in more than one cell type. It's not, just something that's a bit weird. This made me think of the idea of the moonshot program and the Apollo program in the late 60s, early 70s, where the warhead was removed from an ICBM and replaced with a life support system that could take eventually 3 astronauts to the moon and return them safely. And we thought, well, this isn't too dissimilar to these protein toxins that have evolved to actually do something that I personally consider useful, getting a large molecule into a hard to reach intracellular compartment like the cytosol, because that's exactly where the target of ricin is. So we have a number of protein toxins. We have ricin, very well known, cholera, sugar toxin, diphtheria toxin, and anthrax toxin. And all of these large molecules deliver a large molecule to the cytosol. They use different routes, navigating through different processes inside the cell. And we tried a number of these, and the one we settled on was anthrax toxin. And anthrax toxin is quite special because it utilizes an airlock to get large molecules out of this compartment into the cytosol, which means it doesn't have to burst any membranes. You're not causing decompartmentalization. So this airlock is known as an intraluminal vesicle, and it is a function of, it exists to help with the destruction of activated mitogenic receptors like EGFR. So here is our plowshare. Here is our recombinant attenuated anthrax toxin that we've modified. We've removed the warhead, we've gotten rid of the catalytic domain, leaving LFN and protective antigen. Protective antigen oligomerizes to form a pore, which, driven by low luminal endosomal pH, undergoes pre-pore to pore transition and inserts itself into the ILV, the airlock membrane. There's then a translocation event where in the wild, cargo such as edema factor or lethal factor would move over the PA pore into the lumen of the ILV, and that ILV would undergo back fusion, releasing the ILV's content into the cytosol. No membranes are destroyed. So we made recombinant PA protein and also LFN, which is the attenuated lethal factor, non-lethal lethal factor, fused to either PKR or the RNA binding domain of protein kinase R and Saccharomyces cerevisiae gal4. We put these on cells and we were delighted to see, after conducting some quite crude subcellular fractionation, that we could see in the cytosol our cargo molecule here, LFN gal4, and we could inhibit this by removing PA from the system or removing temperature from the system, which is indicating specificity. Because these are toxins, we were also interested in making sure that we weren't going to kill the cell. So we compared that toxicity using a standard MTT assay with polyethyleneamine, which I'm sure we're all familiar with. And you can see the data here. MTT over 24, 48, and 72 hours over the sorts of concentration ranges that we would use to mediate transfection. You can see that we don't actually reach an IC50. And if we look at sublethal toxicity from the perspective of morphology, you can see here with these cells, nuclei, nucleus, that when we stain lamp positive compartments, we don't actually see any changes in these compartments after transfection. This is in contrast with transfection using lipofectamine, where you can see these aggregated late endocytic structures. Okay, does it work? So we strapped some antisense oligonucleotides to gal4 associated with LFN and incubated them with cells with PA protein and compared the antisense effect measured by Western immunoblotting relative to a housekeeper to nuclear affecting our antisense oligos. Nuclear affection here is shown in black, and these ivory bars here are our activity relative or driven by the LFN PA system. And you can see here that the activity with increasing amounts of ASO is more or less equivalent to nuclear affection. This, we saw this also over time. and persisting over time over 12, 24, 36 hours, and in a variety of different cell types. We published this lot, this data set in 2016, and we also could use this system to deliver siRNA as well, which we were very pleased about. But hold on, how can this be? If you look at the structure of the PA pore, there is something called a Phi clamp right at the bottom of the vestibule of the PA pore, which is only 6 Armstrongs across. And that is too small to let through a bit of siRNA, which is about 2 nanometers in diameter. So we were kind of interested in this. We've made sure that our PA protein behaves in a predictable way. We've done small angle neutron scattering on the recombinant PA that we assembled into monomers. And the dimensions here agree with the dimensions published on PDB. However, this does beg the question, could we actually be bursting the limiting membrane of the endosome rather than having our material move through the lumen of the PA pore? So we thought this was a good question to answer. So there was a paper published by Abrami in 2004 from Van der Groot laboratory, where they used siRNA to downregulate a protein called ALEX. And ALEX is a very interesting protein because it regulates this backfusion of the airlock event, which has been characterized as releasing lethal factor and edema factor from anthrax toxin into the cytosol, facilitating the activity of lethal factor here, hydrolyzing MAP kinase. You can see here the data where the target is not hydrolyzed after the siRNA knockdown of the ALEX protein. So we thought to ourselves, could we actually inhibit the formation of ILVs, because we would predict that that would have a similar effect to the siRNA downregulation of ALEX. I'm sorry, is there a way to manually change slides? This is really doing my head in. That would be perfect, thank you. Right, so we need a way of assaying this inhibition of ILV formation. These are from a paper from Bright et al. From Paul Luzio's laboratory. Quite an old paper now, but still lovely. You can see here lots of ILV content relative to cells that have been exposed to hypertonic sucrose. These sucrosomes have increased intraluminal pressure, and that does quite a good job at inhibiting vesicle fusion and also ILV formation. We've also had a go at cross-linking membrane using wheat germ agglutinin, which at higher concentrations leads to these expanded endocytic structures. So we reasoned that this increased osmotic pressure inside our sucrosomes would prevent ILV fusion or ILV formation and should inhibit PA in a similar way to downregulating ALEX. However, we would also be, because of this increased osmotic pressure, increasing the stress on the membrane. So if we were putting PA on there and we were bursting that membrane, we would see an exacerbation of our delivery in sucrose, but not with wheat germ agglutinin, which we were hypothesizing would reinforce our membranes. Next slide, please. Right, so we need some assays to check this out. The first one is a well-published assay which requires the delivery of diphtheria toxin A chain fused to LFN through the PA pore. So we made recombinant diphtheria toxin A chain. Without PA, there's not a great deal of toxicity recorded, even at quite high concentrations of LFN DTA. However, when you add active PA, boom, you kill your cells. Next slide, please. We also have an assay where we are delivering siRNA and measuring siRNA activity. This is in cells stably expressing GFP-fused LAC-Z, and we can measure LAC-Z activity by spectrophotometrically measuring X-gal hydrolysis in lysate. And I've talked about this assay quite a lot in these meetings. Next slide, please. So here are our results. You can see here that we are looking at the inhibition of inhibition. So we got a double negative. Here is cell kill from PA plus LFN DTA, and here is the diminishing cell kill after treatment with wheat germ agglutinin. Well, okay, that still doesn't tell us that we aren't bursting membranes. We see the same effect with siRNA. Next slide, please. However, when we incubate our cells with hypertonic sucrose, you can see the same kind of effect. which would lead us to believe that no, we're not actually bursting the limiting membrane of the endosome, but we are actually having PA translocation activity. Next slide, please. And those sentiments are summarized here. Next slide, please. So if we have PA translocation, can we quantify this by looking at the translocation of radio-labeled, 32P labeled oligonucleotides? So we radio-labeled siRNA against GFP and also STAT3, and you can see the results of that labeling here. We've quantified free label in the system and also the specific activity, and we've looked at the subcellular fractionation and the associated enrichment of markers that tells us our subcellular fractionation is reasonably clean. Next slide, please. So when we introduced our 32P labeled anti-GFP siRNA into HEC293 cells, we could measure the amount of cytosolic versus the amount of membrane-bound 32P siRNA. I will actually draw your attention to this graph or this portion of this slide. These are increasing doses of siRNA. However, the specific activity in terms of CPM and mass is actually going down, and that's represented here. So although it looks like these gray bars are doing something quite strange, you need to remember that there's actually a much smaller amount of siRNA being internalized by the cell. And we can relate this to siRNA activity and excal hydrolysis by the target gene. Next slide, please. We can also see the way that the PALF system compares with a number of other commercially available transfection systems, which are serving here as positive controls. And you can see here membrane versus cytosol in MCF7 cells when we use PA-LFN PKR to deliver our 32P labeled oligo versus oligofectamine and lipofectamine. And that's also been related to siRNA activity by RT-qPCR. So here you can see after transfection with PA-LFN PKR, quite a profound reduction in mRNA. We can also see this manifest as a reduction in protein by Western blot analysis. Next slide, please. We can also use fluorescent microscopy to see Cy3 labeled siRNA in cells. I think you can see here vesicular structures which manifest as LAMP-positive puncta, and it's quite obvious to see here that although there are puncta associated with the siRNA signal, there's also quite a lot of red signal which isn't in puncta. Whether that is due to membrane binding, I think it's probably unlikely because of the difference between the membrane here and the membrane over the nuclei. So that would also suggest another stream of evidence to show that we've got our siRNA into the cytosol using this system. Next slide, please. Right, so I'm going to draw this to a couple of conclusions. Inhibitors of endocytic processes such as wheat germ agglutinin and hypertonic sucrose suggest that PA-poor translocation is needed for the cytosolic localization of LFN-DTA and also LFN-PKR siRNA. Cytosolic radiolabeled siRNA was observed within the cytosol using 2 different RNA sequences and 2 different cell lines, and we could monitor associated siRNA activity via RT-qPCR, via looking at target protein activity and also Western immunoblotting. Next will be to take all of these numbers and relate mass of siRNA per compartment per cell to activity. And that's a story for another day. Next slide, please. With that, I need to thank my fabulous PhD students who generated all this data. Norhan, Hadia, Benedita, thank you all very much. This is their data that I've presented to you today, and thank you for your attention.